Hello and welcome back to Equity Tutors. Today's lesson I'm going to be talking to you about meiosis. So in the previous lesson we went over what mitosis and cell division was, and today we're going to go into a different type of cell division called meiosis. I would really recommend that you go back and make sure that the last lesson is really clear in your head before you try and listen to this lesson, because the stuff we're going to go over today is really based on the steps of mitosis, and if you don't have that foundation then you're going to get very lost. I'm going to go over everything quite slowly to make sure that it's very clear, but remember, go back, listen, pause and revisit this several times until it all makes sense. Again, with this module I would really recommend you looking at the accompanying visual document and also going out to check out our YouTube video, as it will all be a bit clearer once you see some illustrations of what is happening at each point. Okay, so now I've gone over that, let's get started on today's lesson. So if you remember from last week, we talked about mitosis as part of cell division. So when a normal bodily cell divides, it undergoes mitosis. However, we did mention briefly that not all cells in the body have this process, and actually another type of cell division is called meiosis, and meiosis is only performed by gametes. So those are the sperm and the egg, so the two cell types that come together for sexual reproduction. So one of the things that we're going to go over here is exactly what is produced from meiosis. So if you remember from last lesson, during mitosis, you have the parent cell and it produces two genetically identical daughter cells that have the full set of chromosomes. So they have 23 pairs of chromosomes, so 46 chromosomes in total. You have one maternal and one paternal chromosome, remember, and that is very important to keep in mind throughout this lesson. Mitosis produces cells that have the full set of chromosomes, and we call those diploid cells. So I always remember that as D for diploid is the same as double, so each chromosome is doubled. There's a maternal and a paternal chromosome. So for example, you have chromosome 1, you have 1A and 1B. So 1A comes from your mum, 1B comes from your dad. So that's how I like to remember it. However, meiosis produces haploid cells, and it produces 4 haploid cells. So one parent cell undergoes 2 lots of division and produces 4 daughter cells. But these daughter cells are not diploid, they are haploid, so they only contain 1 of each chromosome. So they contain 23 chromosomes and 23 single chromosomes. So there's only 1 chromosome for each number of chromosomes, if that makes any sense. So chromosome 1, instead of having 1A and 1B, there will just be 1A or 1B. So you will only have 1, they don't come in pairs. So in a haploid cell, which I remember is haploid for half, only has half the number of chromosomes. So they only have 1 chromosome for each number of chromosomes, they don't come in a pair. So that's because when the sperm and the egg come together, then you get 1 from your mum and 1 from your dad, and then the somatic cells, which are the normal bodily cells in the offspring, will have a full set of 23 pairs of chromosomes, so 46 chromosomes in total. I know this sounds very confusing, but I'm going to try and keep going over it throughout the lesson, and hopefully it will finally make a bit of sense. So the first thing to know about meiosis is that it contains 2 divisions, and these are split into meiosis I and meiosis II. So meiosis I is pretty similar to mitosis, which is why I said you need to really go over that before you and get that solidified before you go into this lesson today. Okay, so during meiosis I, there are several stages which are exactly the same as in mitosis. So we have prophase, metaphase, anaphase, telophase. So remember PMAT. And meiosis, like mitosis, begins with interphase, where all the chromosomes are replicated. When we start meiosis I, there are double the amount of the normal DNA in the cell. So normal cell has 2 copies of each chromosome, a maternal and a paternal one. And together that makes up 46 total chromosomes, because you have paternal and maternal for each number chromosome. So you have 2 chromosome 1s, you have 2 chromosome 2s, 2 chromosome 3s, etc. After interphase, what you have is each of those will be replicated, so you have a total of 92 chromosomes. So you have what we call the sister chromatids, so an exact copy of each chromosome. At the beginning of meiosis I, the cells have undergone interphase, so each of the chromosomes will have duplicated itself, and now it will be made up of 2 sister chromatids, so those are exactly the same. So 2 copies of each chromosome. So normally we have 46 chromosomes in total, but just before meiosis or mitosis, there will be 92 chromosomes in total. And half of those will be exactly the same as the other half. So there's 2 of each chromosome. So for chromosome 1, there will be 1A, which comes from your mum, and 1A, there will be 2 exactly the same copies. And then there'll also be 1B, which you will have got from your dad, and there will be 2 copies of 1B. So you'll have 4 chromosome 1s, but they will be 2 sets of 2. So when we have the paternal and the maternal chromosomes together, we call those a homologous pair. So they are both chromosome 1, but they are different. And when we have exact copies of the same chromosome, just before cell division, we call that a sister chromatid, and you'll remember that from the previous lesson. At the beginning of meiosis I, we have exactly the same starting material that we do at the beginning of mitosis. So we have double the amount of DNA. And then the first thing to happen in meiosis I is prophase. So during prophase, we have exactly the same thing that happened during mitosis. So prophase has the condensation of the DNA, and then following on from this, we also have the breakdown of the nuclear envelope, and the nucleoluses disappear. So again, if you don't remember this, go back to the previous lesson. Then there is metaphase, and metaphase again is exactly the same. So we have the centrosomes reach the opposite poles of the cell, and the spindle fibres start coming out. And this is where the chromosomes line up on the equator. Now this is where things are going to start to get slightly different. So remember, at this point, each chromosome is made up of 2 sister chromatids. And also remember that there are the full set of chromosomes in these cells. So at this point, the spindle fibres start coming towards the centre of the cell to grab hold of those chromosomes. Before this happens, there is another stage to meiosis which is a bit different. This is what happens in meiosis that differs to mitosis, is this thing called crossover. And crossover is where DNA between homologous chromosomes, so that means like your parental chromosomes, so the one you get from your father or your mother, so I've called them 1A and 1B before, they actually swap bits of DNA. They will line up next to each other and they will cross over, so you will get a bit of chromosome 1A will go on to 1B, and a bit of chromosome 1B will go on to 1A. So what you're left with at the end of this is that those 2 chromosomes you then have are actually completely different to what you have in your normal bodily cells. So this is a way to introduce more genetic variation in the offspring that are produced, because they're not just going to get exactly what their parents have. They actually get a combination of different alleles, which we're going to talk about later in the modules, but this ensures that the offspring and the cells that are produced through meiosis have very different genetic makeup to the parental cells. What we call these new chromosomes that are produced, we call these recombinant chromatids. Remember, that means that they have a bit of, each chromatid has a little bit of maybe another one of its pair. So it will have 1A and 1B, so both chromosome 1 but one you would normally have from your mother, one from your father, they will cross over, and then you will have a new chromosome 1A because it will have a bit of chromosome 1B in it, and vice versa. I know that all sounds very confusing, so really do go and make sure you look at the visual document for this lesson to get that a bit clearer in your head. The other thing that we have in Meiosis I is that during metaphase, when the chromosomes line up on the metaphase plate, the way that they line up can affect what the daughter cells, what chromosomes they have. So I'm going to go over exactly what happens in each stage, but if any of this is unclear, please do comment or give us an email and we will try and make it a bit clearer for you. This is quite a difficult part of the module, so don't worry if it isn't making too much sense when you first go over it. So during metaphase, when all the chromosomes line up on the equator, during metaphase of mitosis, that's when the sister chromatids get pulled apart. So you get one sister chromatid for one cell and one sister chromatid for the other cell. This is where meiosis actually differs. So during metaphase 1, what happens is that the chromosomes line up and they're made of the sister chromatids. But what happens here is that when the spindle fibers attached, they pull the combination of sister chromatids to one pole. This means that there will be different combinations of maternal and paternal chromosomes inherited during Anaphase I. To make that a bit more simple, I'm going to go back to this 1A and 1B. So normally what you would have in metaphase during mitosis is you'd have 1A and 1B, and you would get one copy of 1A and 1B to each of the two daughter cells. What happens during meiosis, during metaphase 1 / anaphase 1, is that you'll get one bivalent, so made up of the sister chromatids. You'll get 1A going to one daughter cell, and you'll get 1B going to the other daughter cell. So then the two daughter cells will have two copies of the same chromosome, but the two daughter cells will have different copies to each other. So one will get one that you had from your mother, one will get one you had from your father. And remember, at this point, they will also have had some crossover, so they'll actually be kind of unique chromosomes to what we normally have in the body. So one of the ways that more variation is introduced into the offspring is that this lining up at the metaphase plate is random, so the assortment of what chromosomes go to which daughter cell is random with each meiosis division. So this is another way that this introduces variation into the offspring, and this is very beneficial, and we're going to go on to that in later modules, why that is so beneficial. Then we have Anaphase I, and during Anaphase I, the different homologs get pulled apart to different daughter cells, but each homolog is still made up of the two sister chromatids. So what that means is that the sister chromatids stay together, but each individual chromosome is pulled apart and they go towards different sides of the cell. And again, what happens is in telophase 1 is exactly the same as before. The nucleus starts to reform and the DNA starts to decondense, and at this point each chromosome has two sister chromatids. So and the two daughter cells are haploid, as they have half the number of normal chromosomes. Remember, at this point the two daughter cells that are produced are already genetically different, so this is the first part of the cell division where different daughter cells are produced. So this differs to mitosis, where the two daughter cells would be the same. At this stage, we get nuclear membrane reformation and chromosomes decondensing, although this doesn't always happen, as they are about to undergo a second round of meiosis, which I'm just going to go into. And also what happens here is cytokinesis, which is exactly the same as what happens in mitosis, where the cytoplasm splits to form two daughter cells. So at the end of meiosis 1, what you need to know is that you have two haploid daughter cells, but that each chromosome at this point is made up of 2 sister chromatids. So they still have double of each chromosome that they do have. And something that can go wrong at this stage is something called nondisjunction. Now, this can happen at Anaphase I or Anaphase II, and we'll go into how this can happen at Anaphase II later on. But nondisjunction is the incorrect separation of the chromosome. So during Anaphase I, this will result in 4 affected daughter cells. And if it happens in Anaphase II, it will only cause 2 daughter cells to be affected because the first one will already have split up. What we call having the incorrect number of chromosomes, we call this aneuploidy, and this can cause many problems, and there are many syndromes that are caused by having the incorrect number of chromosomes. So this can be having too many of one or not enough of one. I'm just going to explain a couple. So there's Edwards syndrome, which is trisomy 18, which means trisomy means there's 3 copies of chromosome 18. So instead of having 1 maternal, 1 paternal, they will have 1 maternal and maybe 2 paternal, or the other way around. Down syndrome is similar and is made from trisomy 21. Patau syndrome is trisomy 13. Klinefelter syndrome is where you have XXY, so you've got 2 X's and a Y chromosome. And then you have Turner syndrome, where you only have it's monosomy X, which means that you only have 1 copy of the X chromosome, and this can only happen in females. So as you can see, the nondisjunction can have major effects on the offspring of the person whose gametes are produced this way. So overall, with meiosis I, the important things to remember is that at this point you get the separation of the homologous chromosomes, and it causes a halving of the total chromosome number, so the cell goes from diploid to haploid. And that extra variation is introduced via crossover and the random assortment of homologs at Anaphase I. And again, if this is not correct, in Anaphase I or Anaphase II, you get nondisjunction, which can lead to aneuploidy. I know that's all a lot to take in, so really don't worry if it's sounding really confusing and that you're not understanding what's going on. Go back, go watch the YouTube video, look at the visuals, and hopefully it will start to set in. Okay, so then let's have a look at meiosis II. So at meiosis II, we have to remember that from the original parental cell at the beginning of meiosis I, that has now split into 2 daughter cells that are genetically different. And then each one of these will undergo meiosis II. So we'll just follow one cell through the stages of meiosis II, but remember that it also has another cell going through exactly the same thing. I've put a diagram that explains this a bit, so I would recommend looking at that so you get an idea of exactly what's happening. So meiosis II is where the sister chromatids are separated. So in meiosis I, the homologs, so the maternal and paternal chromosomes, so the 1A and 1B were separated, but now the 2 copies of 1A will be separated again to form 2 daughter cells from there. So again, this has the same stages as mitosis, so it has prophase, metaphase, anaphase, telophase. So again, you can see why it's really important to get those mitosis cell division really hammered in and clear in your mind before you even attempt to understand what's going on during meiosis. So we then have meiosis II. So meiosis II is again prophase, metaphase, anaphase, telophase, but with a 2 after each one. So during prophase II, the centrosomes duplicate between meiosis I and II, so that there are a new set of centrosomes to release the spindle fibres to pull the sister chromatids apart. And during prophase, the chromosomes condense and the nuclear envelope breaks down if it was reformed after meiosis I, and the centrosomes move apart to the different poles. So that should all be pretty familiar to you. It's pretty much the same during meiosis I and also during mitosis. And then we have metaphase II, and during this stage, the sister chromatids line up along the metaphase plate. So remember, there are no longer homologous chromosomes, they are just for each of the 23 chromosomes that you have in these daughter cells, they will each have a sister chromatid, and these will all line up on the metaphase plate. Then we have Anaphase II. So during Anaphase II, the sister chromatids are pulled apart by the microtubules, and they move towards the opposite poles. And then finally we have Telophase, which is where the individual sister chromatids arrive at the opposite poles and they begin to decondense, and then the nuclear envelope will form around the chromosomes, and now each nucleus contains 23 chromosomes. So at this point you have 4 daughter cells, because this Meiosis II is happening in parallel with the 2 daughter cells that were produced at the end of Meiosis I. Again, this can sound very confusing, so do go check out the YouTube video, or also have a look at the visual document attached to the podcast. Then finally, once we have our 2 nuclei, which are both at this point haploid, they contain 23 single chromosomes, then we have cytokinesis, and this separates the 2 daughter cells into 4 unique haploid daughter cells. So at the end of the second part of meiosis, we come out with 4 daughter cells, and they are all unique. And these can be either the sperm cells in a male or the egg in a female. And then what happens in sexual reproduction is the egg and sperm join together, and then you have 1 chromosome that came from your father and 1 that came from your mother. They come together, and then the offspring will have the complete set of 46 chromosomes, and that contains 2 copies of each chromosome. So each chromosome is a pair, so there's 1 A and 1 B. I know all of that was a lot. Again, go back and listen just to check you've followed everything. And if you do have any questions, don't hesitate to contact us for some more clarity. Following on from this, I'm just going to briefly go over stem cells. Like we looked at in the previous lesson on mitosis, not all cells are able to divide by mitosis, and stem cells can divide an unlimited number of times by mitosis. Most somatic cells, so cells in our body, have a limited number of cell divisions they can undergo, but stem cells can divide an unlimited amount of time. And then what we say is these stem cells are able to form any type of cell. And when they divide, they can either remain, the daughter cell can either remain a stem cell, or it can develop into a specialised cell. And this process of becoming a specialised cell is called differentiation. And so, for example, a hair cell is differentiated different to a cell in your heart. Although they all have the same chromosomes, they are differentiated to different functions. And we will talk a bit more about cells forming tissues and the specialisation of cells later in the course. But for now, what you need to know is that stem cells can divide to remain stem cells or to develop into a specialised cell. And the way we describe stem cells is we say that they have different levels of potency. So there are 3 words we can use to describe them. We can call them totipotent, pluripotent, or multipotent. And I'm just going to briefly go over what these mean now. So a totipotent cell, or totipotency, means that the cell can differentiate into any cell type, and these types of cells are found in the embryo. And totipotent cells can form any cell type in the embryo, and they can also differentiate into extra embryonic cells like the placenta and things like that. We then have, so that's the highest level of potency, so that's the most undifferentiated cell you can find, are totipotent stem cells. We then have pluripotency, and pluripotency is cells that can form any cell type in the embryo, so they're just a little bit more differentiated than the totipotent cells. And then finally we have multipotent stem cells, and these are found in adults, and they can only differentiate into certain cell types. So, for example, we have bone marrow stem cells, and those can form blood cells, but they can't go on and form a heart, for example. So they've gone a bit further down the differentiation pathway, so they can't form all the cells in the body, but they still are not differentiated, and they can form a certain number of cell types within the body. So stem cells are really useful in humans, and you might have heard of them in the news because they have the potential to be really useful for treating disease. And so in an adult, like we said, there are a certain number of adult stem cells, and they're found within certain tissues in the body. So we have bone marrow stem cells, which are used to produce different types of blood cells, like neutrophils or macrophages. And then we also have brain stem cells, and they're used to produce different kinds of neural and glial cells within the brain. These stem cells act as a reserve, so if there is damage, they can come forward and produce more cells to repair that damage. So as I've said, these cells act to repair and replace cells that are damaged in the body. But because of that, they could be extremely useful to treat certain diseases which are characterised by damaged tissue. So we call this stem cell therapy. And this is normally using adult stem cells. And for example, they might donate bone marrow stem cells, and this could be used to treat leukemia, which is a cancer of the blood cells. However, when adult stem cells are being donated, the donor needs to be quite closely matched to the recipient. Otherwise, the recipient might, their immune system might reject the stem cells. So ideally, the patient's own stem cells would be used to treat them, as there's a much lower risk of rejection. So some examples of injuries and diseases that can be treated with stem cells would be hopefully type 1 diabetes, where stem cells could be differentiated into insulin-producing cells in the pancreas. Heart attacks, where they could be used to repair damage to the heart. We could have spinal injuries, so they could be introduced to repair the damage to the nerves in the spine, and so on and so on. However, the greatest potential for stem cells is using embryonic stem cells, and these are derived from the embryo, and they have greater level of potency. They're either totipotent or pluripotent, as we discussed earlier. And because they're less differentiated, they have the potential to become more types of cells. However, because these are coming from embryos, many countries have banned the use of embryonic stem cells, even for research purposes. In the UK, embryonic stem cells can be used for research, but it's very tightly regulated. And you can separate embryonic stem cells into two potencies. So we have totipotent stem cells, which are from the first 3 to 4 days after fertilization, and then we have pluripotent, which are after day 5. And the embryos used in research are often the leftover embryos following in vitro fertilization treatment. So for people that can't have children, or they're not conceiving naturally, they might undergo IVF, which is in vitro fertilization treatment, and they normally produce a lot of embryos, and any embryos that aren't used and implanted and produce a baby they're normally left over and they're not used, but they could be used for research and other treatments. However, because these embryos could turn into a human, many people have ethical objections about using them in research or medicine. So in the exam, you might be asked what the ethical considerations of using stem cells are. And then finally, plants also have stem cells, and we call this meristem. And it consists of undifferentiated cells, and these are normally found in the tissue between the xylem and the phloem. Again, we're going to talk about plant structure later on, but this is a kind of bit of an introduction. And this tissue is known as the cambium, and in the cambium, this is where we find this meristem. So stem cells in plants we call meristem. And in general, the thing to remember is that you find them in the roots and the shoots. So you find meristem in the root tips and the shoot tips, because this is where the plant will be growing. So they need new cells to differentiate and divide. Okay, so I know that was a hell of a lot of information, so don't worry if that didn't all go in today. Just keep listening and go over it again, and hopefully it will eventually sink in.
Thank you so much for listening. Remember, you can access additional content on our Patreon page by searching for Equity Tutors, where we have a second 30-minute lesson every week, plus monthly bonus content. You can also find us on most social media platforms. We will keep you updated on new content, and you can find us there by searching for Equity Tutors UK. Please like, share, subscribe and comment wherever you are listening, and if you're enjoying, please leave a review. Bye!
Bye!